Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I am speaking with Tomasz Kostriezewski, CSO at CN Bio, an organ-on-a-chip company on a mission to transform how human-specific efficacy and safety data is generated in order to accelerate drug discovery pipelines. And today we will be discussing such technologies. Welcome, Tom.
Hi, great to be here.
So to begin with, perhaps you could share with listeners a little about your journey to today, Tom, the passions that drove you to do what you do with CN Bio.
Absolutely. So I've been with CN Bio nearly a decade now. I joined the business at its very early days as it was a young spin-out organisation, originally a spin-out company from the University of Oxford. But very quickly prior to that, I did a PhD in London in molecular immunology, a wonderful department. I also worked in big pharma at GlaxoSmithKline, and always kind of knew that I wanted to work in young, sort of innovative companies developing technologies for life sciences. So when I came across CN Bio and some of the team, I was really excited about what they were trying to develop. And I'd always worked in human relevant in vitro models and how these can be used to predict what happens ultimately in patients. And so, as I said, over the last 10 years, I've been working at CN Bio, trying to develop our organ-on-a-chip or microphysiological MPS technologies, and I'll typically use that term today, which has been a really exciting journey. And I started as a scientist, leading the scientific side of the business, and over the last number of years, I've also taken over the engineering and broader all the technical aspects of the business. So I always sort of refer to myself as a biologist, ultimately by training, but an engineer by osmosis, by working with the great team we have here at CN Bio to develop the microfluidic and really, really intricate bioengineered technology that we have that ultimately enables us to grow really very complex and intricate microtissue models of human organs in the lab.
Thank you, Tom. So if we turn to CN Bio now, and the company has over a decade of experience in engineering these single and MPS systems, if we look at the work it does and the range of products and services that it offers, which includes a benchtop physio mimics OOC, which is organ-on-a-chip range, that can accurately mimic human physiology for more effective preclinical insights. Tom, can you tell us more about industry's need for these new alternative methodologies, or NAMs, and provide background on the capabilities of OOCs?
Absolutely. So if we think about it, first of all, at the highest level, the typical drug discovery process is still pretty much as it was for the last number of decades. We start with broad, high-level screens of large numbers of molecules, and they get whittled down through that drug discovery process against a particular target. And eventually those sort of leading candidates typically get put into in vivo animal models, small animals, rodents, and then larger animals to predict how they will behave in humans. And then those things that are successful will get taken into clinical trial. And the really sort of difficult and unfortunate fact is that clinical trial success rates are really very low. The efficiency of the drug discovery industry is quite shocking, in that 90 to 95% of clinical trials do not succeed. And so this is costs a huge amount of money, huge amount of time, and ultimately we're failing patients in that sense that there are wide ranges of disease states that we want to try and treat, and we simply can't. And there are lots of activities across the drug discovery industry to change this. From developing new, more human-specific molecules. So rather than using traditional chemical entities, we're using biological entities, whether they be proteins, antibodies, oligonucleotides, or even cells. But what is also really important that we have is that when we test these in the drug discovery process, we don't necessarily test them in those sort of slightly more traditional manners. So it's all well and good developing a drug that works fantastically in a mouse model of a human disease, or a larger human model of a human disease. But if it doesn't work in a human, well, it doesn't have a lot of value. So a number of years ago, probably the last 15 to 20 years, there's been this real strong movement of developing NAMs, new alternative methods, of which In vitro models is just one. So there are other things such as in silico, chemical, in silico, predictive, computational approaches. But in vitro, can we really culture human tissues or human cells as microtissues to predict what human responses will be to these drugs? And that's the typical premise of what we're trying to do at CN Bio. And by building organ-on-a-chip technology, we're basically creating a microenvironment to grow human cells. Typically, we put these in dishes that are standard, that are flat, that are not representative of human physiology. And these cells really struggle to grow. They don't have the same form function that they would do in a human body. So by building these more intricate bioengineered platforms, it allows us to, culture multiple cell types together. We can culture them in a three-dimensional environment, and we use microfluidics to bring fluid flow. And what all of these things do is ultimately allow us, as I say, to create these microtissues that represent human organ and tissue function at some state. So that then means we can then screen new drugs, whatever entity they may be, chemical or biological, to better predict how they will behave ultimately when they go into a patient.
Thank you. So you mentioned the applicability to certain disease areas. I was just wondering, could you exemplify particular areas that are being revolutionized with this technology currently?
Yeah, so again, I'll start broad and then narrow down. The technology is used both to model disease states, but is also used to predict and model off-target effects in toxicity, and also important studies for ADME, ADME, and DMPK. So we can really use these tools across the drug discovery and development processes. So in a tox-based assay, we would culture a healthy tissue, and then obviously we can screen to look for adverse effects of a particular compound. And what's important is that we can measure we're trying to measure the clinical endpoints that would be measured for that particular tissue type in a clinical trial. So for example, if we take the liver, there are some classical enzyme levels that get elevated in the human blood. Those are those same enzyme elevations we look at in our liver model of liver toxicity. And so therefore the data we can generate from these platforms is really translational. And we can use these models, as I say, in the ADME space. We can look at to predict what appropriate doses could be used for in vivo and ultimately clinical trials. So how do we predict the drug exposure of a particular drug in a human patient? Using some of these chips. And then, as you said, we can really generate potentially a wide range of different disease states depending on some of the materials and some of the methodologies that we've generated. So there are models of a number of different diseases, particularly some metabolic diseases now have some very well-established models. Some neurodegenerative or some neurological diseases, some rarer neurological diseases have been modeled very nicely in these types of devices, and a whole range of oncology-based models have been developed using organ chip technologies. And these can be very powerful and more predictive than the standard approaches of modeling these disease states in, say, more simple in vitro approaches with cells in dishes, or in animal models that often can be very difficult to predict or difficult to model certain disease states.
Thank you. So if we take OOCs together, why do some of these technologies stand out over others?
So I think what's important is, and this goes with any sort of new technology that's being developed, is that what we're trying to do is develop something that's very innovative and very cutting edge, and therefore the practical usability elements are just as important as the sort of the core fundamentals of does it do what it says on the tin. So the number one thing is, can it produce translational data? That's always the first and foremost what we're focused on. If it can't do that, if it doesn't have some improvement over the predictive value of more simple approaches, well, then actually there is little value in us doing it. So we always start with that, and we compare very closely the data that we generate, ultimately, if we can, with data from clinical studies. That can sometimes be challenging to translate, so then we'll maybe take one step further back and we'll compare to the data generated in vivo studies. And really, we're looking for this collation of data. And what's important is we're also looking to thickness of data. So a typical in vitro assay with cells, basic cells cultured in sort of static dishes, is actually that only really simple endpoints are often assessed. So you might look at, say, the amount of ATP the cells have, or the presence of one or two certain markers. With these much more complex in vitro models that we're generating in our MPS organ chip devices, we're really looking for a wide range of different endpoints, all from the same microtissue that we generate. So we might look at the transcriptomic profile, the proteomic profile, we might look at secreted clinically relevant biomarkers, we might look at a range of histology of the microtissues, and really then bring all of this data together to ultimately allow us to compare between different treatment regimes. But as I said, we have to also do this in the context of the user. So the more successful technologies, such as the ones we've been developing at SynBio, well, they can be adopted by anybody in any lab, and we can get them integrated. They don't take very much training, that we can set scientists up in just a couple of hours. With the technology, it doesn't need an expert. It actually doesn't need an engineer to run these platforms. It just needs a biologist. Some of the organ chip technologies that have been developed by others are incredibly elegant, have an incredible amount of intricate approach, but they really struggle with that tech transfer. So actually, the use of them cannot be scaled across the industry very easily. And in parallel to that, these technologies also have to have throughput. Any drug pharma company wants to be able to screen hundreds or thousands of different chemical entities or biological entities to determine the one that's going to potentially be successful. And so these technologies have to have throughput. And so the types of devices that we build and have the capacity in a single study by a single scientist to screen tens or even hundreds of compounds across a range of doses, where other platforms that have been developed have a throughput of sort of n of 1. And so therefore, you might be able to run 12 chips together in an experiment, but that only gives you an n of 12 rather than an n of, say, 200 or 500. And that can be really limiting to the broader use of these technologies. So our whole has always been balancing these three things. We balance ultimately translational data and biological predictivity is always at the top, but it has to be usable. It has to be scalable to be used in anger by the pharmaceutical industry.
Thank you, Tom. So going back again to SynBio itself, the past year has been a time of rapid growth for the company. Notably, in December 2023, SynBio's physiomimic system and liver model, which you mentioned earlier, were used to provide crucial data to support Innopharm's IND submission for INI-822, which is an industry-first example of an organ-on-a-chip provider's data supporting clinical progression of a drug for metabolic fibrotic liver disease. However, I was wondering if you could comment more generally on the regulatory progress and market adoption when it comes to OOC technology.
Yeah, so the regulators have taken a very active role in trying to understand how this technology works now for a number of years. Probably some of our first interfaces with regulatory agencies were sort of all the way back in the early 2010s, and they've really accelerated over the last number of years. That comes in a number of forms. We see varying approaches from some of the different regulatory agencies around the world, and this sort of goes somewhat in parallel to the broader market adoption. So to go a little bit deeper, we started working with the FDA in the US back in 2017 in a more formal sense. We've been sort of collaborating with them ever since. And what that means, in the first instance, they were just really trying to understand what these technologies were, what type of data they could generate, and whether this data would have any value. And that has now started to mature further, and there are now activities ongoing across a range of different groups to look to more formally qualify or validate these types of technologies in a very particular context of use for particular regulatory assays. And so in the longer term, that will mean, and we can envision that regulators would potentially stipulate or mandate the use of these technologies. But that isn't quite the case today. But what's really important is that regulators are trying to encourage pharma companies across the board that they will accept data right now, today, using this type of approach. Depending on the scientific question they're looking to answer, the amount of validation of the particular approach might vary. So as the decisions get ever more critical, go closer and closer to the clinic, then the potential scrutiny around that particular question that they're asking might get a little bit higher. But they are now seeing a much wider range of use of these technologies in regulatory filings. But they're still relatively in its infancy. The vast majority of the use of this technology is sort of the first 80% of the drug discovery process to allow pharma companies to make internal decisions about progressing molecule A versus molecule B, and not so much about whether a molecule is absolutely safe to progress into clinic. But that is now starting to evolve. This type of data that organ-chip technologies can generate is used as part of a broader sort of risk assessment process. The data can be submitted alongside more standardized approaches that have been sort of long well accepted. And so the regulators and particularly the FDA are really excited about these technologies. They really see the value that they can now bring. And they're actively encouraging organizations like ours to look to do these more formal qualification packages. And lots of these things will happen over the years to come. And as I said, that broadly mirrors the sort of market adoption that we've seen. So certainly when I joined this industry, nearly a decade ago now, we were just talking to those very sort of early adopters you classically see in any market, the real kind of real innovators who want the most cutting edge tech. We obviously continue to work with those for a long time, but we've now much broadened out into the more mass market of users within pharma. And in particular, a sign of that is that we're now working very closely with a wide range of global CROs, and the CROs are coming to us to understand this technology, how it fits in their workflows alongside those more standardized approaches that they're very familiar with. So really, we see that market adoption growing. I think, again, we must be also honest that it's still in its earlier stages. We certainly don't see every single drug using this technology yet, but we see in the years to come, it will become commonplace. And certainly from a personal perspective, it's very rare that I meet someone from pharma these days who doesn't know what an organ chip MPS is and some of the value it can bring. So really, it's now about how much value can it bring, how broadly can it be used, and which technologies will be adopted the fastest. And certainly we see that multiple technologies will be adopted across the industry for different contexts of use, different needs, different scientific questions. And it's really just about the scale and the pace that goes at. And there is top-down drivers. We've heard from FDA Modernization Act, and that similar things are coming from other regulatory agencies too. That they want to drive this replacement of typically animal approaches for sort of the 3 R's benefit, and how that can happen. They are looking to make that happen more quickly, and so there's a sort of drive from a number of fronts. But ultimately, it's always focused on patients and ultimately focused on the best quality science to get the data that people need to make the right choices about which molecule to take forward with the greatest confidence into a clinical trial.
Thank you, Tom. It's been a pleasure.
Thank you.
And so that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous instalments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on iTunes, Spotify, Amazon Music, Podbean, and pretty much wherever you get your other podcasts. Just search for pharmaphorum and press subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us as well on X or Twitter and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.